It is October 1921. A 35-year-old doctor is standing at the absolute pinnacle of his career. He is a Boston Brahmin, born into immense wealth and privilege and educated at the elite halls of Harvard College. He has just been named the Chief of Medical Service at Harvard's prestigious Huntington Hospital. He oversees state-of-the-art medical laboratories and is on a path to dominate American medicine for decades to come. But lately, something is catastrophically wrong. He takes a Wednesday off to serve as an usher at his brother's wedding, but he can barely stand. His muscles ache with an inexplicable weakness. Worse, he is gripped by an unquenchable thirst. He drinks gallons of water and is constantly urinating, yet his mouth remains dry, his body starved for more. As a physician, he knows exactly what these symptoms suggest. He does not wait for a consultation. He goes straight to his own hospital laboratory and tests the sugar levels of his own urine. The chemical reaction turns the liquid a deep red, indicating massive amounts of sugar. He knows immediately he has a disease that has plagued humanity for all of recorded human history, and he will be dead before he reaches the age of 40. Welcome to the dawn of science-based medicine. I am Professor Jim Brody. The man who tested his own urine that fateful Wednesday was Dr. George Minot. Before 1922, a diagnosis of diabetes mellitus was an absolute guarantee of a short, painful demise. The human body, entirely unable to process glucose, essentially starved itself to death, while its bloodstream drowned in toxic levels of sugar. Desperate, Dr. George Minot sought out nation's leading diabetes specialists, another Boston physician named Dr. Elliot P. Joslin. At the time, the brightest minds in medicine had no idea what exactly caused the disease at a molecular level. They only knew that the origin of the failure was located deep within the abdomen, in a spongy organ called the pancreas. Dr. Joslin's philosophy was harsh, but reflective of the technological limits of the era. He believed the pancreas was simply overworked and damaged. His treatment was focused exclusively on extreme dietary restriction, hoping to give the organ a rest. A starvation diet. When patients inevitably fell ill and died, Joslin often blamed the patient for cheating on the strict regimen. Here's what you need to know about blood sugar levels. There's a normal range. As you eat, your blood sugar goes up higher. The body responds by releasing insulin, which tends to push it lower. If you don't have insulin, you go higher and higher. George Minot was way up here in the 400s. At that level, you can die. It takes months. If you have too low of a blood sugar, you can die quickly, within hours. Minot was a towering man for his time, standing 6 feet 1.5 inches tall, but he weighed a mere 135 pounds that day. Joslin immediately placed him on a starvation diet. He was allowed just 1,250 calories a day. A week later, as the blood sugar refused to drop to safe levels, Joslin slashed his intake in half to only 525 calories a day. To survive, George Minot had to have great discipline. He carried a heavy metal spring kitchen scale everywhere he went. He meticulously weighed every single gram of food before it entered his mouth, recording the mathematics of his own starvation. By February 1922, Minot's weight had plummeted to a skeletal 122 pounds. The starvation diet worked, temporarily, by keeping blood sugar just low enough to prevent patients from slipping into ketoacidosis and comas, but it came at a physical cost. Hospital wards across the country were filled with emaciated children. The air smelled of acetone, the chemical byproduct of a human body consuming its own fat and muscle tissue just to keep the brain alive. To stop this suffering, science had to find the missing link, and to find it, they had to look back 32 years to an aha moment after a scientist watched a well-trained dog repeatedly urinating on a laboratory floor. The year is 1889. The location is Strasbourg, a city on the France-German border. Oskar Minkowski, a brilliant and meticulous surgeon, is engaged in a fierce academic debate with his colleague, Joseph von Mering. They are arguing about how the mammalian body digests fats. Von Mering believes the pancreas is essential for digestion of fats, but Minkowski disagrees. Oskar Minkowski was scientific royalty. His brother Hermann was a world-famous mathematics professor who developed the description of space-time used by Albert Einstein. Oskar himself was renowned for his surgical precision. He was perhaps the best surgeon in the world at the time. This was a great debate in the 1880s, and the debate is whether the pancreas is essential for life like the heart or non-essential like the appendix. Minkowski thought he could just remove the pancreas. He could answer the question by simply observing whether the animal survived. But a total pancreatectomy is a very difficult operation. The organ is hidden deep in the abdomen, wrapped around the intestines. If a surgeon leaves even a microscopic fraction of the pancreas tissue behind, the organ continues to function, ruining the data. But Minkowski's technique was flawless. He completely removed the pancreas from a healthy dog, sutured the abdominal wall, and waited. The dog recovered, lived, and initially appeared perfectly normal. Minkowski had demonstrated that an animal could survive without the pancreas. The debate seemed settled. But then, the dog, which was thoroughly house-trained, began urinating constantly all over the laboratory floor. Minkowski did what all great scientists do when confronted with an observation like that. He said, that's interesting, and investigated. Minkowski collected a sample of the dog's urine in a pipette and ran a chemical test designed to detect the presence of sugars. The result was staggering. Minkowski knew what it meant immediately. He had accidentally induced severe diabetes in this dog. This was a monumental breakthrough. Minkowski had created the first true animal model of diabetes. In biomedical research, an accurate animal model is the crucial first foothold in solving any disease. Without a way to reliably replicate and study a condition in a controlled laboratory setting, scientists are just guessing in the dark. The scientific community suddenly realized that the pancreas was not a useless appendage. It was actively producing a mysterious, life-saving secretion that we call insulin today. The solution to diabetes now seemed incredibly obvious to the medical community. If removing the pancreas causes the disease, then introducing a healthy pancreas to a diabetic patient should cure it. Doctors rushed to the local slaughterhouses and acquired raw cow and sheep pancreases and fed them directly to dying patients. In 1910, one desperate doctor fed a patient several raw pancreases daily for an entire month. The result, absolute failure. Nothing happened. The sugar levels remained lethal. Grinding up the pancreas and injecting the tissue directly into patients also failed catastrophic. To understand why this simple tissue transfer failed so spectacularly, we have to look at the microscopic architecture of the organ. Here's what you need to know about the pancreas. It has two functions. One, shown in these blue tissues, are the digestive function. These tissues produce proteases, proteins that destroy other proteins and feed them here into the small intestine, the digestive system. The second system within the pancreas is shown in these purple tissues, the islets of Langerhans. That produces insulin. Insulin goes into the bloodstream. In a living body, these two functions are kept strictly separate. But the moment you take a whole pancreas from a slaughterhouse and grind it up in a blender, you mix the two assembly lines. The corrosive digestive proteases are freed. They act like molecular scissors, instantly shredding, digesting, and destroying the delicate insulin protein before it can ever be extracted or utilized. This was the great biochemical bottleneck of the 20th century. To save millions of lives, someone had to figure out how to separate the fragile insulin from the corrosive proteases. By 1920, the greatest scientific minds in the world had mostly given up. The extraction was deemed biochemically impossible. Meanwhile, Dr. George Minot, surviving on 525 calories a day, was rapidly running out of time. The intellectual center of the world was firmly rooted in Europe at that time. Every single science Nobel Prize awarded prior to this point had gone to European researchers. But the answer to this problem wouldn't come from a grand institute in Berlin or Vienna. It would come in the dead of night, in October 1920, to an unknown small-town physician from rural Canada. His name was Frederick Banting. He was a farm boy who struggled with spelling, failed out in his first year at university, and pivoted to a medical degree. After serving as a triage surgeon in the blood-soaked trenches of World War I, he returned to the small town in Ontario and opened a solo medical practice. He had zero research experience. In the first month of his practice, he saw exactly one patient, a man looking for a prescription for alcohol to bypass prohibition laws. Deep in debt, Banting took a side job as a lecturer at the local medical school. While preparing for a class on the pancreas, he read an obscure medical article by Moses Barron. The article by Moses Barron reviewed a particular pathology. In those cases, there was a blockage here, stones formed. Whenever they saw those stones, this blue tissue was all destroyed, leaving only the endocrine tissue that produces insulin. Late one night, Banting jolted awake. He grabbed a piece of paper and scrawled a bizarre, highly dangerous surgical idea. Banting hypothesized that if you operated on a living dog and artificially tied off the pancreatic ducts with a ligature, the corrosive digestive cells would back up and die off over the course of several weeks. The insulin-producing cells, however, would survive this blockage. You could then safely remove the degenerated pancreas and harvest pure insulin, entirely free from the destructive proteases. Frederick Banting's 2 a.m. dream was brilliant. It would prove that the insulin protein could be surgically isolated in a living animal without being digested by its own enzymes. But Banting was about to slam into a massive engineering wall, because while performing a complex duct ligation surgery on a single dog might yield enough insulin to keep another dog alive for a few weeks, how could you possibly scale a 6-week surgical procedure to save the lives of millions of dying humans? The cure was real, but mathematically, the extraction process was going to limit the cure to only a few.